So we're looking at reproduction in cells, and remember that depending on if one parent contributes the DNA or two parents contribute the DNA, we call this asexual or sexual reproduction. Think of it as if you have a pen, that means you have one pen. So in asexual reproduction, there's only one parent contributing the DNA. The offspring are genetically identical to the parent. There's less genetic diversity, but it allows for a lot of offspring to be created in a short amount of time. In sexual reproduction, there are two parents that contribute the DNA, and this allows for a lot of diversity or differences between the offspring. And that's going to be good because it's going to increase the survival of those offspring. Now, because we have two different cells that have to be joined to make this new individual, we're going to have a different way of dividing the cells in order to have only half the number of chromosomes. And this is where meiosis comes in. So this is a form of division that allows for sex cells being made or the gametes being made. And so remember that the gametes are the egg and the sperm. And so they are going to be haploid or have half the number of chromosomes. So that way, when they join during fertilization to form the new individual, this zygote is diploid and it has twice the number of chromosomes, or has two sets of the chromosomes, one from their mom and one from their dad. So we're going to look at that process now. Here is an overview of meiosis. It is a form of cell division, but it's only for forming gametes. We start out with diploid cells that have two sets of chromosomes, and we're going to end up with 4 haploid cells. So this is telophase, so it hasn't split yet, but we'll have 4 haploid cells. And this is going to happen slightly differently in males and females. At the end of this, in males, we'll have 4 viable sperm cells, but in females, because of the amount of cytoplasm and all of the organelles, there's only 1 viable egg and 3 what we call polar bodies, and those will be reabsorbed by the cell. But the DNA is distributed equally amongst those. It's just only 1 will be viable. So let's look at this process. We start with interphase. We're still going to duplicate or make a copy of the DNA. But in meiosis 1, prophase 1, these 2 copies, one from our mom and one from our dad, that each have their copies, so we have 4 of the same type of chromosome, they're going to form a tetrad. Tetra means 4. And so if you've ever played Tetris, it's a game that has different combinations of those 4 boxes. So in a straight line, in a zigzag, and a square. So these 4 chromosomes are going to cross over, and crossing over looks something like this. Watch how they break apart, reattach, and they make new combinations for the next generation of individuals. And so that's one of the advantages of crossing over, is that you have new possibilities or new combinations that can occur in the offspring. After prophase 1, during metaphase 1 of meiosis, notice they do line up in the middle, but they're not single file like we saw before. In metaphase 1, they're in pairs. In anaphase 1, the entire pair gets pulled to each side. And again, forgot to say in the last slide that you can see where crossing over happened and we have the new combinations. In telophase 1, we see that the cell's pinching in and it's starting to go through cytokinesis, but we have entire chromosomes. In prophase 2, the nuclear envelope starts to disappear. Now notice, and starting in prophase 2, what's happening is in 2 cells, not just 1. In metaphase 2, they are lined up in the middle. And again, this time single file. In anaphase 2, the centromere breaks and they split to either end. And finally, we have 4 haploid cells. So we can see that in meiosis 2, we end up with these 4 haploid cells. And again, that's going to look slightly different in males and in females. We'll go over some of the differences between mitosis and meiosis, as well as